Today we're going to present about coliform bacteria for bioremediation of waste hydrocarbon. Research objective. The major objective of this study was to investigate the hydrocarbonolastic potential of coliform from local domestic sewage sample.
Next, we will look at material and method. The first one is counting and categorizations of coliform in sewage. Also, we use the conventional dilution plating and the coliform selective medium, oxine methylene blue agar. The isolation were further categorized according to their potential for acid and gas production from lactose fermentation at 37 Celsius and 44 Celsius. Then, we will comparing the sequence. Basically, it will amplify by polymerase chain reaction or known as PCR with a standard protocol. The second method is hydrocarbon utilization potential of coliform. As we can see, the mineral medium had the following composition and the pH was adjusted at 7. In this method, in order to study the range of pure hydrocarbon that could be utilized by the individual strain, a common inoculums was prepared for each strain. Culture were incubated at 30 Celsius for 15 days. The third method is hydrocarbon consumption by coliform. It was measured by gas liquid chromatography (GLC). Around 100 ml of mineral medium in 200 ml flask was supplied with 0.3% crude oil or pure hydrocarbon. Each flask will be incubated at 30 Celsius with 200 rpm on electric shaker for 2 weeks. Next, we determine hydrocarbon uptake by coliform. In this experiment, 2 strains of E. coli, one from domestic sewage and the others from wheat straw, were investigated by using transmission electron microscopy (TEM) for intracellular hydrocarbon inclusion. The other method is oil removal in sewage batches. 100 ml of freshly harvested raw sewage were provided separately with 1.0 and 5.0 gram portions of crude oil. At a time 0 and 5-day interval, culture were harvested for quantitative oil and coliform analysis. After all the experiments are being conducted, the statistical analysis will be done and the mean value with the standard deviation values were calculated. ANOVA was used to differentiate between the means of tested parameters.
Moving on to the next section, which is the results. This section is divided into 4 parts. Without further ado, let's move on to the first part. Local coliform bacterial strains. Based on the results obtained, the coliforms consisted of Enterobacter hormeaceae subspecies O'Harae, Klebsiella verrucola, Klebsiella pneumonia, and E. coli. As we can see from Table 1, the similarities between the 4 isolates and the standard strains in the gene bank database were high, between 99 to 100%. For the acid and gas production, all isolates give positive results except for the Klebsiella and Enterobacter, since they did not produce gas at 44 degrees Celsius. Oil and hydrocarbon utilization by the coliform isolates. As we can see from Table 3, Enterobacter is the coliform that exhibited the highest percentage of hydrocarbon consumption. Pseudomonas tatsiri was also analyzed as a classic hydrocarbon utilizer for comparison. Cytological evidence for C16 uptake by E. coli. Figures A and B show the transmission electron micrograph for E. coli cells from domestic sewage that have been grown in N-hexadecane-free medium and N-hexadecane-containing medium, respectively. On the other hand, figures C and D indicate the TEM images of E. coli cells from wheat straw. Last but not least, self-cleaning of oil sewage by indigenous coliform. This part shows the graphs of crude oil, C18 and phenanthrene added to the freshly raw sewage and steadily removed after 20 days of incubation. For the first graph, the red dotted line shows the remaining proportions of 3% crude oil was 70%, with the total number of coliforms increased to 26.5 times 10 to the power of 6 CFU per milliliter. The second graph shows 45% of 1% crude oil remain after 20 days with 52.7 times 10 to the power of 6 CFU of total coliforms, while the remaining C18 were 66% with total coliforms of 18.9 times 10 to the power of 6 CFU per milliliter. The last graph shows 55% of remaining phenanthrene and the total number of coliforms increased to 15.8 times times 10 to the power of 6 CFU per milliliter.
Now, we will proceed with the discussion. In this section, it is divided into 4 sections, which are local coliform bacteria strains, oil and hydrocarbon utilization by the coliform isolates, cytological evidence for N-hexadecane C16 uptake by E. coli, and self-cleaning of oily sewage by indigenous coliforms. 4.1 Local coliform bacterial strains. Based on the result obtained, the CFU rating indicates that the total viable bacteria found in the 3 raw sewage is high. Furthermore, 5 different coliforms that were identified show the similarity of 99 to 100% indicates that the isolates are on the same branch, which means that they belong to the same species or the strain were located on a very close branch. Moreover, the result for several species of coliform that illustrated the negative production of gas at 44 degrees Celsius is due to the inability of the species mentioned to grow, which is the same as the expected generic identities. 4.2 Oil and hydrocarbon utilization by the coliform isolates. In this section, the result illustrated that the coliform has the potential in utilizing the hydrocarbon. This can be observed from the ability to grow on the solid medium containing crude oil vapor as the sole source of carbon and energy. Also, the selected strain considered has the potential for hydrocarbon utilization because of the ability to grow in various individual N-alkenes and individual aromatic hydrocarbons, toluene, naphthalene, phenolphthalein, and biphenyl. The successful growth of the strain indicates that they produce monooxygenase and dioxygenase, which is like the hydrocarbonoclastic organism. Apart from that, the metabolic pathway of the coliform also is similar to other hydrocarbonoclastic microorganisms. Thus, it can be concluded that the result shows that the coliform has the hydrocarbon-clastic potential.
Next, cytological evidence for N-hexadecane uptake by E. coli. The EM image in Figure 2 show that the cell cytoplasm of hydrocarbon incubated cells, in contrast to that cells harvested from nutrient broth, was enriched with less electron-dense areas of varying dimensions. The measurements showed that those inclusions occupied roughly 50% of the total cell volume. Earlier researchers also described similar intracytoplasmic hydrocarbon inclusions in classical hydrocarbonoclastic bacteria incubated in media containing N-hexadecane. Cells incubated in the presence of C16 also showed clear morphological deformations. Self-cleaning of oily sewage by indigenous coliforms. Figure 3 shows further that Enterobacter was the coliform that exhibited the highest increase in number, which may reflect a main role of this coliform in oil consumption. However, the significant increase in numbers of coliforms parallel to the significant decrease in the concentration of the remaining oil provide an experimental evidence for major contribution of indigenous coliforms to self-cleaning of oil in sewage. Other hydrocarbonoclastic non-coliform bacteria indigenous to sewage are also probably involved in the measured crude oil removal. The finding implies that the indigenous domestic sewage microflora is capable of controlling potential oil spills in the waste product. Furthermore, raw sewage is an accessible, cost-effective source of hydrocarbonoclastic microbial cocktails that can be used for bioaugmenting oil-contaminated areas.
This group should be particularly effective in cleaning oil spills in sewage as those organisms are autochthonous, or in the other word is indigenous. Conditions prevailing during the conventional sewage treatment probably bioenhance the coliform's potential for oil bioremediation. Aeration should bioenhance the spilled oil bioremediation as the initial step of microbial attack on hydrocarbons is catalyzed with oxygenases or hydroxylases. Lastly, the nitrogen fertilization guaranteed in sewage for the hydrocarbonoclastic coliform communities. It is because during sewage treatment, nitrates and other nitrogenous compounds are continuously produced. This finding means that the domestic microflora of indigenous sewage is capable to control the oil spills in these waste products. The coliform bacteria in domestic sewage, probably in other environmental materials, are effective hydrocarbon biodegrading microorganisms. The bioremediation of waste hydrocarbons using coliform bacteria also gives some benefits, such as it is a cost-effective and an accessible source of microorganisms.